Welcome everybody to our today's webinar. It's part of a series within our VIRTIGATION project. And today we want to talk a little bit about cross protection, how we can combat plant diseases with mild virus isolates. My first slide is missing, just a little bit of housekeeping rules. I will give a short presentation on the background of cross protection and how different mechanisms may behind this effect that we can observe. And Elise will give you a little bit more details on what it takes to take mild isolates to the market and use them actually for plant protection. And we will take questions after the talks, but via chat only. So please, if you have any questions, address them in the chat. So I mentioned the VIRTIGATION project. VIRTIGATION deals with emerging viral diseases in tomatoes and cucurbits and looks for implementation of mitigation strategies for durable disease management. And this project is funded by the European Union's Horizon 2020 Research and Innovation Fund with almost 7 million euros. And as you can see, the project has been running for quite a while and will come to an end this year. We have an international consortium of 25 partners from 12 countries. And the consortium is coordinated by Evert van der Schuren from the University in Leuven in Belgium. And you can see from the little pictures here that we have a huge range of research institutes and companies that are involved in this project. So the challenge was that new aggressive viruses are spreading in greenhouses and fields, and they are threatening the multi-billion value chain of tomatoes and cucurbits. And in this project, we focused on two very important virus groups, the whitefly transmitted begomoviruses, represented by tomato leaf curl nudelli and tomato yellow leaf curl virus, as well as the mechanically transmitted tobamovirus, tomato brown rugose fruit virus. And due to their rapid transmission, these plant viruses are difficult to control and cause complete crop loss in affected areas. And in the case of whitefly transmitted viruses, producers often use chemical pesticides to fight these viruses. And this may expose growers and consumers to pesticide residues. And at the moment, only limited organic solution exists on the market to combat these viruses. And the aim of this mitigation project is to develop rapid and long-lasting solutions to combat emerging viral diseases in tomatoes and cucurbits. And this is being implemented through 6 specific objects. First of them, knowledge sharing and engagement of stakeholders in research activities. And that's exactly what we are doing today with this webinar. And there will be more webinars to come, so watch this space. Develop robust diagnostic tests, quarantine measures, and identify ecological factors driving disease outbreaks, understanding plant virus vector interactions, and under Objective 5, the development of integrated pest management solutions. Cross protection is seen as one possible solution. But we also want to identify and pyramid natural resistances to viral diseases and vectors, and train the tomato and cucurbit value chain. And if you would like to have more information, you can go to our website, virtigation.eu, and you can also find us on many, many different platforms in social media. And at the end, Elise will also provide you with a link for our stakeholder registration form, where you can sign up and get some information about the progress of our project. So, cross protection, let's dig in. What is cross protection? Simply saying, it's a primary infection with a virus that prevents a secondary infection with a challenging homologous virus. And this has already been described for almost 100 years ago in 1929, when McKinney observed that there are different mosaic diseases available and they are restricting each other's. And early mechanism hypothesis involved the formation of immunizing substances, difficult word, like we have in our bodies. But of course, plants do not have an immune system, strictly speaking. The formation of antibody, competition for replication materials, or the occupation of virus-specific replication sites. I'm going to present you the more validated hypothesis. So one big part is the coat protein mediated protection. And it was seen that the coat protein of a protective strain can partially coat the challenging strain's RNA and therefore prevent infection. And this has been shown for Brome mosaic virus, where had Brome mosaic virus replicase extract, and when the coat protein of this virus was added, the RNA synthesis was inhibited. But if a coat protein from a completely different virus, tobacco mosaic virus, was added, the RNA synthesis was not inhibited because these two viruses are too distantly related. It was also shown for tobacco mosaic virus that the coat protein of a protective strain could prevent protoplasts from infection with a challenging TMV strain. But this only worked when the challenge occurred with viral particles and the protective strain prevented the uncoating of the TMV viral RNA, and therefore the challenging strain could not infect the protoplasts. But if you used TMV purified RNA, then the challenging strain could infect the protoplasts. And this demonstrated that the coat protein was indeed important for cross-protection to occur. However, cross-protection was also seen for, let's say, molecules which do not possess a coat protein. Viroids are single-stranded circular RNA molecules, and they are not coated by a coat protein, but yet we can observe cross-protection. And Horst in 1975 could see that Chrysanthemum chlorotic mottle virus, a viroid, could protect a plant against a similar Chrysanthemum chlorotic mottle viroid strain. And therefore, the conclusion is that the coat protein may play a role in cross-protection, but is probably not the only mechanism involved. RNA silencing was described almost 30 years ago, and what it means is that a primary infection may activate the RNA silencing machinery of a plant and therefore prevent a challenging strain to infect that plant. And it has been shown that the application of double-stranded RNA molecules that are targeting a challenging virus can induce RNA silencing and therefore protect these plants. And we took the example of Bienenkommen mosaic virus, and there were double-stranded RNA molecules targeting the NIB and the coat protein genes, and this exogenous application of plants prevented the successful infection with a challenging BCMV strain after 5 days. Just to give you a brief recap what that means. So each plant virus doesn't matter if they have an RNA genome or a DNA genome. At one stage at their replication cycle, they form double-stranded RNA molecules. And this is quite unique to these viruses, and it can be recognized by plants, by different enzymes that then chop these double-stranded RNA molecules into smaller pieces and therefore can either prevent the infection or can produce even more double-stranded RNA molecules to facilitate a signal amplification. And when I started working on plant viruses, we thought, wow, this is a cool idea. Let's take a cucumber mosaic virus mutant, which cannot express a silencing suppressor that would compromise the plant immune system. And this mutant that cannot express the to-be silencing suppressor will be a good inducer of RNA silencing. And it should induce a strong systemic signal and therefore be a very good protective agent. And we found out that, yes, the cross-protection worked quite well when we challenged the viruses on the same leaf. But when we challenged the protective strain with the parental strain on different leaves, the protection didn't work so well. And this indicated that there was not a very strong systemic silencing signal. And furthermore, we observed that, yes, the cross-protection worked also in silencing deficient Arabidopsis mutants. And therefore, we concluded that RNA silencing may play a crucial role in cross-protection, but again, is probably not the only mechanism involved. What researchers observed is a phenomenon called spatial separation. So if you have different strains of the same virus, it seems that they can exclude each other from infecting the same cell, spatial separation. And Edgar Meis and his PhD student, they tagged viruses with different fluorescent proteins, and they found out when we use a potivirus and a potex virus with different labels, they can infect the same cells. But if we use a potivirus with two different labels, they cannot infect the same cells, and there's a clear spatial separation between, let's call them here, protective and challenging strains. But the mechanism behind it could not be elucidated. And this spatial separation has been observed for many different virus species, potex and potiviruses, apple-latent spherical virus, CMV, AMV, and also tobamoviruses. This spatial separation is also sometimes referred to as super-infection exclusion, and that has also been observed for many taxonomically different viruses with different genomes, also in the human and animal virus world. And it seems that for each virus species, there are different players being involved. For example, the P28 protein of turnip-crankill virus, the M protein for Sonchios yellownet virus, or the P33 protein for citrus tristeza virus. And what is common that these proteins inhibit the viral replication cycle at one stage or another, be it by transmembrane transport, by different locations within the replication sites. And it seems, therefore, that for different viruses, we have different mechanisms of cellular exclusion that may occur, but we all refer to them as the phenomenon of cross-protection or superinfection exclusion. Just a little bit of background, what we are talking about. But cross-protection has been quite successfully been used in many cases to protect crop losses, for example, so in the yellow mosaic virus in cucurbits, papaya ring spot virus in papayas, and citrus tristeza virus in citrus plants. And last but not least, pepino mosaic virus in tomatoes. And this is a very nice success story, and Elise will talk a little bit about what it takes for a mild virus to become a cross-protecting agent that can commercially applied to protect crops.
Thank you, Heiko. I hope you can all see my screen at this point. So I will continue with the talk. As you can imagine, now that we have the background, cross protection is quite an interesting strategy to also apply as a plant protection tool, so to protect plants from viral infection. But of course, before you can come from the R&D point of view, proof of concept, to a product that is actually applicable in the market, of course, there are some requirements that a mild virus isolate has to meet before it can actually be developed into a vaccine for cross-protection strategy. So that is what I will talk to you about first. So first and foremost, we're talking about mild virus isolate. So the first prerequisite is, of course, that your isolate is mild in all conditions. So what does that mean? It means that you have no or only mild symptoms on the leaves and on the fruits. There should not be any impact on quality or yield. And it also means that your isolate is consistently mild under all conditions. So in all varieties, all climate conditions, all cultivation conditions. So a lot of viruses are known to be influenced by these different conditions. So it's very important to do your research and evaluate if the phenotype of the isolates that you are studying for a cross-protection strategy indeed fits the bill and shows mild symptoms even under all these varying conditions. A second very important one is, of course, then also the genetic stability. So your isolate also has to be genetically stable. So most plant viruses have RNA genomes that are replicated by RNA polymerases that they check the sequence, but they don't do a very good job. So they don't have proofreading activity. So that means that when your virus is replicating, that it can be that a mistake is made in the transcription of the genome. So that means that mutations do happen when viruses are replicating. So that's something to definitely check. The genetic stability of the isolates has to be studied and compared with aggressive variants. It can also differ between isolates. It can be very dependent on selection pressure by environmental conditions, but also selection pressure exerted by the host plant in any context. So again, when developing a cross-protection strategy, this is something that definitely needs to be checked because isolates with high mutation rates are, of course, more challenging for vaccine production and quality control. If you have an isolate that is not very genetically stable, you cannot guarantee to your customer that at each point they will be receiving exactly the same sequence, exactly the same mild isolate. So this is a very important one to also check. Biological fitness, so we're talking about mild isolates, but they should not be weak. So what does this mean? Your protecting isolate needs to result in a fast, homogeneous, and systemic colonization of the plant. So when you apply the isolate, it should be the case that your entire plant becomes infected, that there is an equal distribution of the vaccine, so the isolate concentration throughout your plant, but also throughout your crop. So if you're planting in a greenhouse, that all your crops have the same concentration of virus present, and they should be maintained in all plant parts during the entire cropping cycle. So the mechanisms that Heiko discussed based on RNAi and coat protein mediated mechanisms, they do need continuous and homogeneous of viral RNA. Otherwise, it can be that if your plant is not fully protected, if it does not have the correct concentration of virus, it can be that it does not offer 100% protection against an aggressive isolate that might try to infect your crop in a later stage of the crop cycle. Then lastly, of course, viral population study. What this means is your isolate should protect against dominant viral genotypes. So again, you need to be aware of the situation in which you are applying your mild isolate. What viruses are present? What other isolates, what other genotypes are present? And will my isolate actually protect against the other genotypes that is present? Am I applying the correct strategy for the virus that could be threatening my crop? So this is actually a continuous study, so it needs to be continuously monitored, as we have seen in the past that viral populations can shift. So you need to be aware of the population before you start, but you need to continuously keep updating this also to see that your information stays current. And yeah, as I mentioned, the efficacy against dominating circulating genotypes is, of course, crucial there as well. As you can imagine, based on the information that Heiko gave at the beginning of the talk, cross protection is very limited to closely related viruses. So the homology between the protecting isolate and the challenging isolate, challenging dominant population rather, should be high enough that your mild isolate actually confers protection. Some special points of attention there. You need to evaluate the risk of diffusion to other non-target plants. So in the case of tomato, there are crops that are very closely related, such as, you can see this very nice picture of a potato plant on the right-hand side. So they are closely related, tomato and potato. If you are applying a strategy in tomato, you need to know, does my strategy also affect other crops? And if it affects other crops, is it damaging to other crops? Similarly to other plants in the ecosystem, weeds, for example, can act as host virus reservoirs for a lot of different viruses. So even if you applied a mild isolate strategy a couple of years ago, it can be that the weeds are still retaining it. If you plant another crop and there is a risk for that crop to become infected, and it turns out that there is a danger, that is, of course, a big problem. So you need to be aware of these risks, and they should be really non-existent or very, very minimal to other non-target plants. Be aware of mixed infection. So this also lies in line with being aware of your virus population, taking it a bit broader than, of course, the strategy that you're applying. Different viruses can, of course, interact with each other, and mixed infections can differently influence host responses. So depending on what specific isolates are present in the country that you're applying the strategy, or in the crop that you're applying the strategy, interaction can happen. And again, effects of this should be minimal. Also, different climate and growth conditions can influence this interaction. Different varieties can affect this interaction. So these are all things to be aware of and to keep in the back of your mind when you are developing a vaccination strategy. When developing a mild isolate into a cross protection strategy, of course, the production process needs to be controlled, standardized, and needs to have extended quality control. So I mentioned already a couple of things that are needed for your virus, so your mild isolates, before it can be developed into a cross protection. So it's, of course, very important that you keep doing these checks also as a quality control, specifically with regard to genetic identity and stability. Of course, this needs to be safeguarded, that if you are developing a mild isolate into a cross protection strategy, your isolate needs to be the same. It needs to be the same that you are delivering to your customer every time. So it needs to be checked that this is the case. Your mild isolate needs to be pure. So you need to be able to ensure that presence of other pathogens is excluded. It has to be a standardized viral concentration and standardized viability. and standardized composition also, of course, of the product. The vaccination strategy, of course, also has to be optimized under practical conditions and should be easy to apply. This means standardized application methods and correct dosing in order to ensure that your mild isolate is being applied in the correct way to offer protection. And of course, also well-defined timing of application at a specific plant growth stage. Finally, maybe it's already became a bit clear while I was listing the requirements, but cross-protection agents are, of course, plant protection products. So this whole list of requirements is one thing, but there are also many different procedures and legislation for authorization of plant products, plant protection products applicable. And within the EU, it is particularly Plant Protection Regulation 1107-2009 that is applicable. What this means is that the registration of your cross-protective agent as a plant protection product also safeguards the efficacy, makes sure that you are getting the same product, a qualitative product that is also safe to use and efficient to use each time that you receive it. And this, of course, ensures user and consumer protection. So yeah, that concludes my part of the talk. I already mentioned some information about the VIRTIGATION project. So what we talked about here was a bit more general about cross protection. Of course, Heiko also mentioned there will be a couple of webinars coming up. We're doing a webinar series where we will cover different topics of relevance to the VIRTIGATION project, sharing general information and also results, I think, in some talks. So yeah, if you're interested in the project and would like to receive regular updates, you can always scan the QR code, fill in the form, and register as a stakeholder to keep updated about our VIRTIGATION project. And I think that is the final slide. So you can still find the QR code up here if you want to scan it. And then I think now we have some time for questions. So thank you very much for listening to our talk. And if you have a question, shoot.
Yeah, thank you very much, Elise. So sorry for those people who probably expected the next TOBRFV product on the market, but I think Elise made it very clear how long it takes from scratch to develop a marketable product for cross protection, even though it's a really interesting and cool concept. Yeah, this project VIRTIGATION was funded for 4 years, and overall, if I calculate everything together, it took you almost 15 years for pepinomosaic virus to deliver some answers to the growers. So would you expect that legislation or the registration process might be a bit faster these days, now that we have the pepinomosaic vaccine as an example? Or do you think that the legislators and registrators will be hesitant to allow a potential TOBRFV vaccine on the market?
Mm-hmm. I think the legislation is there for a reason. I'm not sure. I mean, of course, we quite recently changed from Q status for on QP status, right, for TOBRFV. So I think that does give a bit more room to a cross-protection product for TOBRFV also becoming an option on the market. But I think the legislation is there for a reason. Is very important that we keep following it. So I don't think there should be any exceptions. I think the pepinomosaic virus vaccination story shows that it can be a very good strategy indeed to protect, but if you have the data to support that. And I think it's important that we keep doing that, right? That we keep having these checks in place to ensure that whatever cross protection strategy comes onto the market is indeed efficient, safe, robust, stable. So I think that's important that we keep checking that. I think Marlene has a question. Marlene, you can ask your question in the chat.
So I think what you presented also is, I think, a very important key message, that this is one tool in the box. So you need to observe your viral population all the time to address your needs. And I think if we should have tolerant or resistant tomato varieties, that is one thing. Keeping hygiene in the greenhouses is another thing. Using a potential TOBRFV vaccine is a third thing. And so probably all these different tools all together make sense for a sustainable protection strategy for your crops. Marlene, can you type your message in the chat or send it by WhatsApp?
It's also possible. Yeah. No, indeed, I think cross protection it's an interesting tool to be further explored, but I think it's also why you mentioned it also for vertigation. We are researching IPM strategy, so it's one of many tools in the defense against the virus that could be possible.
I just got the notification that the chat is not working.
Oh, it's not working. Okay, then I will stop and have a look at that.
Oh, it seems for David now it's working to type in the chat. So yeah, Guadalupe can also not type.
No, but I see that Marlene typed something now, so.
Okay. Yeah, sorry for the technical difficulties. This is our premiere. So there will be many more webinars coming up in the next few months and weeks, and I hope that we have solved the technical issues by then.
So Marlene's question was, can you discuss the use of different strategies like resistant cultivars and cross protection in one environment? So I think that can be a very interesting strategy. Yeah, it's also something that we would like to research in the vertigation project. Of course, to use both in the same strategy still implies that you need to have some kind of level of infection in your crop, right? So it will not be feasible in a resistant cultivar that completely excludes your virus. It's more something that could be combined with a virus that allows some sort of basic level of replication still, where you have a crop that is already resistant, so already shows less symptoms or no symptoms, that is even further protected with a cross protection isolate, so a mild isolate that will even further protect the crop. So I think this could be a very interesting strategy to apply in future as well. That could work very well in combating virus damage.
Yeah, Marlene, I think we need to distinguish here the resistant varieties that will not allow viral replication in the plant, and therefore the plant cannot be infected by TOBRFV, or if we have tolerant plants where the virus can infect the plants, but the impact on viral load and or yield is neglectable or not visible. And that could be a good combination to combine that one with mild strains. So if you have a plant that is already, let's say, a little bit stronger against fighting off viral diseases, and you can then protect it with a mild strain of the virus throughout, then we could use that probably in areas where you have a high contamination level, where it will be very difficult to clean, let's say, greenhouses and the surrounding environment. Yeah, I hope that this answered your question. Are there any other questions? I cannot see anything.
So I see a question, is topography a priority in the VIRTIGATION project? So I think Heiko, you explained it quite well at the beginning of the project, right, of the presentation, right? That we have two different viruses that we are studying in the project. So we have on the one hand the Tolsian DV, on the other hand topography, and I would say that definitely makes topography a priority for this project as well. It's one of the two main pillars that our project is built up on. So definitely something that we are working on in the VIRTIGATION project.
And also at many different aspects. So starting from disinfection strategies from soil towards the cultivation bags, over cross protection as potential strategy, as well as looking for genetic resistance or genetic tolerance towards the virus. Again, if you're interested in all the different work packages, you can find the detailed information on our website, and you will also find information on the published results when they are being published or made available to the public. Of course, after 4 years, we are starting to get the first results, but not all of them have been published yet. So yeah, also sign up for the stakeholder register form, and then you can get information also via the newsletter.
Yes. Then we have a question from Ruti. Any chance we can use cross-protection against DNA viruses? I noticed all the work on cross-protection is on RNA viruses.
The method of delivery is the limiting factor here.
I think so too, yeah.
I think because the DNA viruses are vector transmitted, and Pepino and TOBRFV are both very easily transmitted mechanically, and this makes them, of course, perfect candidate for targeted inoculation. But if we come up with a delivery method for DNA viruses that would not involve, let's say, agrobacterium infection, then it could also work quite well for DNA viruses. But since they are vector transmitted, I don't think it's very easy to build up a whitefly population with your mild DNA virus strains and release them in the wild so that they can infect and pre-humanize the crops. This is, I think, the little problem. And we used infectious clones of DNA viruses. Again, then we have the GMO label, which comes into place. And even if you use purified virions and particle bombardment of plants, it's something that at the moment is not suitable for the masses, to my mind. But if we have a good invention of a delivery tool, I cannot see why it wouldn't work for DNA viruses as well.
No, indeed, I think the important factor there is the delivery. So it needs to be mechanically transmissible for ease of application, as we are not very keen on sending out infected insects to vaccinate our crop. I think that's not very not something we want to do. So that's probably the limiting factor there. Then we have one more question of Marlene, higher chance of resistance breaking. And I think, Marlene, you mean that in case resistance cultivars and cross protection are combined in the same environment, right? Yes. Yes. Yeah, it's I think it depends on how your strategies are also being applied, on how it's managed, on how how your isolate is contained. I think in case of a thinking about neighboring cultivations, Marlene says so. We mentioned a couple of times that a good vaccine candidate needs to be genetically stable. Indeed, if your isolate is breaking resistance, it's indicative of an isolate that is not very stable, that has a high mutation rate, that can mutate to overcome the resistance. And it would also, in case of resistance breaking, imply that the strategy is being combined with a crop that does not allow any kind of replication. So it's tricky, I think. Heiko, I don't know if you have any thoughts about this.
I just mentioned it under the umbrella understanding plant virus vector interactions. So of course we want to know what makes the virus aggressive, which part of the genome is responsible for the aggressiveness on certain crops or within certain varieties. But also we want to know what makes mild isolates mild. And similarly, we want to know if we have resistance, which part of the viral genome is responsible for the breakage of the resistance, and how can we take all this information together to develop protective agents that are not falling under the GMO legislation. So of course it would be very nice if we could use our infectious clone, for example, and we know where to manipulate the genome in order to make it a very nice cross-protecting agent. But unfortunately in the EU that would not be allowed to take to market. And so that political decision limits us scientists, of course, to maybe looking for more targeted and easier solution. But yeah, we couldn't mention everything here today in this webinar. So stay tuned for the upcoming series, and maybe you will hear more about the TOBRFV plant interactions.
No, for sure.
And I think we need to conclude. I just got a little warning that the time is up. So thank you very much for joining us for today's webinar and the interesting questions. And yeah, we would be very happy if you turned in also for the other series, for the other webinars. And as Elise has pointed out, if you have any questions, contact us, contact the consortium, and we are happy to answer your questions.
Yes.
So thank you very much for now, and thank you, Elise, for organizing all the participants.
Thank you, Heiko, and I wish everyone a very good rest of the day then. Hope to see you in our other series. All right.
Thank you.
Bye-bye.
Bye-bye.